Good morning. Today I'm going to present our preliminary work on exploration and spatial abilities of invasive bank voles in Ireland. Whether introduced into a completely novel habitat or slowly expanding their current range, the degree to which animals can efficiently explore and navigate new environments may be a key component of survival, ultimately determining population establishment and invasion success. Biological invasions are the product of a multi-stage process, which requires the invader to successfully negotiate a series of sequential and selective filters. To invade successfully, individuals have to navigate their way through all stages of the introduction process, where each stage poses its own set of challenges. And invasive animals arriving in a new environment lack the up-to-date knowledge necessary to locate and recognize resources that are key to their survival and reproduction. Like, for example, local knowledge about food sources, nesting materials, and opportunities, as well as local dangers like predators, poisons, and climatic exposures. Not all species spread beyond the site of introduction, but studying those that do can reveal a great deal about a species' range limits and how it will respond to environmental change. In addition to morphology and physiology, behavioral traits contribute to many successful animal invasions and range expansions, because they are in general more labile traits, and so more likely to match local environments. Behavioral differences between native and invasive populations have been frequently recorded, and there is also evidence that in species still expanding their range, individuals at the front of the invasion show differences for conspecifics in the established populations in a whole range of traits. Differences among conspecifics in exploration, dispersal, and risk-taking tendencies might also promote the accumulation of certain phenotypes at the expanding invasion edge, causing spatial sorting. Individuals with dispersal-enhancing behavioral traits, such as high levels of risk-taking, spatial orientation, and then predicted to be more common at the expansion edge than in these long colonized populations, both because they enhance an individual ability to find food and shelter in novel environments, and because these traits increase dispersal rates. Here we aim to address the question of whether exploration and orientation are associated with invasive spread, making use of a rare occasion where the expansion edge can be pinpointed, using an ongoing range expansion of bank voles in Ireland. Voles from Central Europe were accidentally introduced to Ireland about a century ago, during a major building operation. A source population was then established at the west coast, and expanded into an island with very few other small mammals to share it with. Voles have been estimated to expand their range about 2.5 kilometers a year, and are currently midway through the colonization of the Emerald Isle. In our study, we measured bank voles' exploration tendency and risk-taking propensity by means of repeated dark light and open field tests, where we measured the animal's latency to emerge from a safe dark shelter into an illuminated open area, and then how they explored this space. Spatial orientation was quantified using a modified version of the standard laboratory radial arm maze that is commonly used in cognitive studies of rats and mice. The first version of our setup was used to test eastern chipmunks' spatial learning abilities directly on the field and without prior handling, and we modified this original maze to adjust it to bank voles' necessities. In the laboratory version of this test, food is the most common reward, and animals are often asked to remember which arms they have already visited and so where the food has already been eaten. Here, the reward we used was the possibility to leave the maze. This is why we conducted the test at the exact site of capture for each animal. They would leave the maze and be in the very spot where we had originally trapped them, and were free to go home. And this is how the test worked. The vole was placed into the first arm of the maze and left to explore the rest of it. When it had visited all arms but one, we opened the last arm, and whenever the vole entered it, it would find it rewarded by an open exit. We then measured latencies to enter and leave the maze, as well as the number of revisits to each arm, meaning how many times the animal would enter a previously visited arm. And this is what we found. In the open field test, individuals at the expansion edge had much longer latencies to emerge from the shelter, which we interpret as being more risk-averse, and longer latencies to start exploring the central, more exposed part of the arena, compared to voles in the source population. In the radial arm maze, voles at the expansion edge not only took longer to enter the maze than conspecifics in the source population, but they also took longer to leave it. Once they found a way out, they would take a while before actually leaving the last arm of the maze and regain freedom. As for the number of revisits, voles at the edge did more revisits to previously visited arms compared to vole in the source population. And lastly, the latencies to emerge from the two tests were correlated, and so weakly were the latency to explore the inner part of the open field and the number of revisits, with voles that took longer to explore the open field area, revisiting more often already explored parts of the maze. So to sum up, there seemed to be a non-random sorting of individuals between expansion edge and established source population. But contrary to our expectations, bank voles at the expansion edge were more risk-averse and took longer to start exploring both the open field and the radial arm maze, compared to conspecifics in the source population. They revisited unrewarded arms of the maze more and waited longer before leaving it. Now, voles are a very heavily predated taxon, and they constitute the staple of the diet of many predators, both aerial and terrestrial. Taken together, these results suggest that for this heavily predated upon small mammal, a careful and thorough exploration strategy might be favored when expanding into novel environments. This is of course a very first step, and we need further information on other aspects of the ecology, cognition and life history of these animals, as well as data taken along the entire expansion gradient to better understand this unusual and timid invasion. Finally, let me say thanks, first of all, to my co-author, Professor Jana Eccard, who did all the groundwork to bring us both to Ireland, to the local experts that welcomed us there, and the university and funding agency that made this work possible. Lastly, thank you very much for listening.